So I'll start off with, TEJASHRI, Purohit-Sheth.
Yeah, nice job. It's actually pronounced TEJASHRI Purohit-Sheth. So thank you for giving me the opportunity to speak today, and I've really appreciated all of the excellent presentations that have preceded this session. And I also wanted to shout out to the patients and the caregivers who shared their very touching stories with us earlier today. So I'm going to talk about long-term patient follow-up. What is it? What are our recommendations with respect to this? So I'll provide some general considerations for long-term follow-up. We'll talk about risks of particular gene therapy products, as well as what should you take into account for risk assessment. And then we'll touch upon preclinical and clinical considerations. So my talk is basically a summary of our published draft guidance in July 2018. This is a long-term follow-up after administration of human gene therapy products. And this is a revision of the earlier guidance from 2006. So what is long-term follow-up? So it's monitoring for adverse events for an extended period of time for subjects that were in clinical studies after the active protocol pre-specified follow-up period, as well as for patients who've received gene therapy products post-marketings. Now, depending on the risk characteristics of the product and what we understand about the gene therapy product, not all gene therapy products will require long-term follow-up, and we'll get into this in the subsequent slides. So why do we need it? So as was discussed earlier, gene therapy products, they are intended to achieve a prolonged or permanent effect, a therapeutic effect. So as such, long-term exposure may result in unpredictable or unexpected delayed risks to the patients who receive gene therapy. What types of potential risks exist? Malignancy, impairment of gene function, autoimmune type reactions, reactivation of latency, and infection, and persistent infections. And all of these are predicated on the type of vector that is utilized in the gene therapy product. So what are the considerations? So you want to look at the specific product characteristics. Where is the gene therapy product targeted to go? What available preclinical or clinical information do we have that may inform the risk profile of the gene therapy product? So characteristics of the gene therapy products that are considered to be an increased risk for adverse effects months to years down the line include the integration activity of the GT product. Is it a genome editing product? Is there a prolonged expression of the transgene? Is there a potential for latency, as may be seen with replication-competent vectors such as herpes virus? Or, latency with herpes-type virus. And then also establishment of persistent infections when you think about vectors such as Listeria. So this is a table that has been taken from our draft guidance, and it basically goes over the propensity of commonly used gene therapy products and their ability to modify the host genome, which is something that is taken into account critically when we think about risk of gene therapy products and delayed adverse events. So when you break that table down a little bit, the products that are considered more risky with respect to delayed adverse events include integrating viruses or vectors such as gamma retrovirus, lentivirus, and then herpes virus, which is capable of latency reactivation, and then products that have genome editing capability. Products that are considered generally at a lower risk of delayed adverse events include plasmids, poxvirus, adenovirus, AAV. But the caveat here is if these products have been modified to somehow allow the ability of transfer of genetic elements, then they may actually be considered at greater risk. This is a framework that we have outlined in our draft guidance, which takes you through a few questions as to whether your product or a gene therapy product poses a risk of delayed adverse events. So moving on from product characteristics, which are certainly important to inform the risk of a particular gene therapy product, we also want to look at preclinical considerations. Preclinical data is really important for us to understand the localization, distribution, and persistence of the gene therapy product, as well as to get an understanding of what sort of on-target and off-target effects may exist. When one is designing studies, preclinical studies, to assess for localization, distribution, and persistence, some of the things that we think are really important to consider in designing such studies are, one, try to use the gene therapy formulation that is to be intended for use in humans, as well as the route of administration, evaluate both sexes, and then try to correlate product persistence and localization to any adverse effects that may have been noted. Characterization of the kinetics of the gene therapy product is very important, and the time of sacrifice of the animals needs to be taken into account. So we recommend that sacrifice is done at the peak gene therapy product detection, as well as at later time points to get an understanding of the clearance of the gene therapy product. And part and parcel of collecting animal data is, how is the tissue being collected and analyzed? So commonly examined tissues should be evaluated, as well as additional tissues based on the tropism of the gene therapy product, the route of administration, etc. And it's very critical when you're looking to assess vector persistence, distribution, that the assay methodology is both quantitative and sensitive, such as PCR. And that assay should be able to detect the vector sequences in both animals as well as humans. So moving on to clinical considerations, when you're looking at the need and the extent of long-term follow-up, you want to look at, what are the goals of the long-term follow-up? Who is the subject of the long-term follow-up? The clinical trial population? Are you looking post-marketing, doing long-term follow-up in that population? Then you also want to consider duration of long-term follow-up. Not all products will require the same duration of follow-up. So that needs to be considered. And then elements of long-term follow-up, which we'll get into in subsequent slides. And then informed consent was discussed earlier, but we also require informed consent for these long-term follow-up studies. So the goals of long-term follow-up, basically you want to identify risks of gene therapy products that are delayed months to years after exposure to the gene therapy product. You'd like to mitigate the risks, so if you identify issues early, perhaps treating, mitigating that risk, and then also get an understanding of, what is the persistence of the gene therapy? How long is it there? How long is it in the body? So for the long-term follow-up population, basically all subjects who have been enrolled in a clinical trial who have been exposed to gene therapy comprises the long-term follow-up population. And when you are considering characteristics of the population that is going to be followed in the long-term follow-up, you want to consider the life expectancy based on the underlying condition that has been treated, assess for comorbidities, and then look at other agents such as radiation or chemotherapy that perhaps have adverse events as a result of exposure to these agents. So duration of follow-up, long-term follow-up, should be sufficient to assess the delayed adverse events. And this is based on, what are the characteristics of the product, what sort of in vivo product persistence has been noted, what is the expectation for duration of transgene expression, and then the in vivo product characteristics that have been identified based on clinical study results, as well as animal data. You also want to consider the nature of the exposure, meaning, what was the target organ, target cell that the gene therapy product was delivered to. You also want to consider the anticipated time of occurrence of the delayed adverse events. So you want to ensure that you are following them long enough so you're able to detect adverse events that may occur 10, 12, maybe even 15 years after exposure to the gene therapy products, such as malignancy. And you also want to consider expected survival rates and known background rates of survival in the study population. So when we think about how long the long-term follow-up should be, for integrating vectors and genome editing products, we recommend 15 years. For AAV vectors, we recommend up to 5 years. And then certainly a risk-based approach may be considered for vectors that are capable of latency, and then products that have long-term expression, but they do not necessarily integrate into the host genome. For the long-term follow-up, we really request that there is a dedicated clinical protocol that really goes into and pre-specifies detailed patient visit schedules. What is the monitoring plan? Also, the protocol should document and specify the sampling plan, when blood or tissue will be collected, the methodology that will be utilized to assess perhaps vector sequences, and then what are the clinical adverse events of interest. Those should be also considered and documented in the long-term follow-up protocol. Because once the patient or subject is out of the clinical study, you're going to deliver them back to their primary care provider or wherever they were living, and it's very challenging to ensure that the same clinical study procedures, monitoring procedures that were in place are going to be implemented once they're back with their healthcare providers. So if you have a long-term follow-up protocol and a dedicated protocol, specifying all this upfront is critically important. Providing perhaps templates for the non-investigator caregivers on what should be collected, when it should be collected, what times sampling should be done, etc. And of course, for us to really understand the risks of delayed adverse events, accurate case histories are critical. So elements of the long-term follow-up plan, they should go into, and I touched upon some of this, but really talk about how you are going to detect these adverse events. How are you going to collect the data? And if there are adverse events, the protocol should specify that these will be submitted to the FDA as part of the IND safety reports. They will be discussed in the annual reports. And then, as appropriate, clinical protocol amendments may need to be submitted. Perhaps there's an emerging risk that needs to be assessed, and so the protocol may need to be modified for that. When will physical examinations be conducted? And then also you want to evaluate and specify GT product persistence. So when it comes to procedures, what would we recommend is collected or done? So the first 5 years, we really would request that the long-term follow-up protocol specified the details of the monitoring plans, what information will be collected, so histories, physical examination, what labs will be collected, reporting of adverse events, and the case history should also pay particular attention to collect and procure information that may perhaps even confound our ability to interpret some of the adverse events that have been noted. So, for example, if somebody has received exposure to a mutagenic agent. You want to know that. That's important information for us to understand. And then also to collect information on emergence of new medical conditions of interest. Knowing how these gene therapy products work, you want to look for malignancy, worsening of neurologic conditions or hematologic issues, rheumatologic conditions, autoimmunity type issues. And so all of this is recommended to be evaluated and collected during the first 5 years. Now, in the subsequent 10 years, if there is no emerging issue or no significant concerns have been identified, then we recommend that subjects be contacted, perhaps remotely, at least once a year. It could be phone, it could be a questionnaire, it could be an office visit with a healthcare provider as appropriate. And so I think in the interest of time, I was a little more ambitious, but I covered some of the stuff already. And so I'm going to move on to informed consent. Informed consent for a long-term follow-up protocol should contain the basic elements that we have in our Part 50.25 CFR regulations. But additional considerations with informed consent should include, how long are the patients or subjects expected to participate in this? What are the foreseeable risks? What tissue, what blood samples are going to be collected? And then you want to ensure that whatever anticipated adverse events are put in the informed consent so the patient can actually understand them and know what to actually look for. If they have something and they don't quite realize that, this could be a side effect of my gene therapy, you want to make sure that all of this is taken into account and put into the informed consent document. They also need to provide informed consent for collection and storage of tissue or blood samples that may be done in the context of the long-term follow-up. And if in the event of death, that is something that should be in the informed consent, but a discussion about autopsy during the informed consent process should be discussed, where providing consent for an autopsy would be very informative to understand the risks of a particular gene therapy product. And so a lot of the discussion that I've had so far has been in the context of clinical trials and following these patients or subjects for a longer period of time following the completion of the active portion of the clinical trial. But you also have to think about, what about once a product is marketed? Then what? So what are the considerations after licensure of a gene therapy product? So at the time of the biologics license application, we discussed with our applicants the need for a post-marketing pharmacovigilance plan. And that plan, depending on the risk profile of the product, may include just routine surveillance, or it may include just submission of reports for serious life-threatening unexpected reactions. It may include registry, may also include a required post-marketing observational clinical trial that is designed similar to the long-term follow-up protocols that we've discussed. And then depending on the risks of the product, a risk evaluation and mitigation strategy may also be required. So I thank you for your attention, and if I have accomplished the goal of notifying you that there is a long-term follow-up guidance that includes all of this information, then I have achieved my objectives. Thank you for your attention.